This video is Chapter 5 Notes, Part 1, all about the cell membrane and osmosis. The cell membrane is what goes around the outside of a cell. It is three-dimensional around an entire cell, and it allows the cell to exchange things with its environment. A cell needs to get a lot of things from its environment. It might need to get oxygen, carbon dioxide, sugar, amino acids. It might need to release things such as oxygen, carbon dioxide, or waste. So the cell membrane is the barrier through which those particles have to pass. The cell membrane keeps everything inside the cell separate from what is outside of the cell. The cell membrane begins with an introduction to this idea of surface area to volume ratio. This is a huge idea in AP Bio. The idea of how much of a cell is exposed to the environment will allow it to do different functions. When I talk about surface area, I'm talking about the perimeter of a cell. The cell on the left has a small surface area. If I took that red line that I drew and laid it end to end, it would be so long. If I took this red line that I'm drawing now and laid it end to end, it would be significantly longer than the one that I drew on the left. So that is surface area. How much is on the perimeter, the outside of the cell? Volume then has to do with how much is inside of the cell. So volume has to do with this area on the inside. Even though these two cells have very different shapes, the volume of what's inside the cell is actually pretty much the same. If you imagine, like if I had a cup and I filled up the cell on the left and I filled up the cell on the right, the same amount of liquid would fit into each of those cells. So even though my surface area was very different, the volume is the same. And that is where this ratio comes into play, surface area to volume, which is often abbreviated like so. In biology, it is best to maximize the surface area to volume ratio, meaning it's best for a cell to have a huge perimeter with a smaller amount of stuff on the inside. Different cells have different functions, and by looking at their surface area to volume ratio, it can tell me a little bit about the function of a cell. If there is a particle outside the cell, like let's say these green particles are food, and these cells need to obtain that food. If that food particle is moving into the cell, on this cell on the left, there's only so much area over which that green particle can float into the cell. Whereas this cell over here on the right has way more surface area, and thus there's more locations over which that food particle could enter the cell. So the cell on the right has an advantage because it has more area over which it can get the food inside. However, it is not best for cells to just be absolutely gigantic. We don't know of any cells that are really big enough to see much without a microscope. So the reason that cells are tiny is because it's all about this ratio. Once the food particle gets inside the cell, it's going to have to float because food is not alive, so the food is not going to be like swimming. So the food particle needs to float by random motion to get where it needs to go. So it is not best for the cell to be absolutely giant because then there's no way that the food particle could ever reach the center of the cell. So it is best for a cell to have a big surface area so it can take in lots of stuff, but then a small volume so that the particles don't have to float very far once they get inside. So cells want a large surface area to volume ratio. Here is a big example of an elephant. Elephants have these huge ears. It maximizes surface area. That allows the elephant to exchange and lose a lot of heat to its environment so that the elephant does not overheat. Shown here are Asian versus African elephants. A distinctive feature is the size of the ears. African elephants tend to live in much hotter climates, so they have huge ears to dissipate and lose heat, big surface area. Asian elephants have not quite had so much pressure from natural selection to lose heat because Asian elephants don't live in quite as hot of climates, so their ears tend to be smaller. So a big surface area will maximize exchange. We see that with a lot of organisms that live in the desert. They have these huge ears. Yeah, maybe it helps them hear predators, but really the main function is to lose heat and keep body temp cool. So the big ears help to lose heat. It's an example of big surface area. Same like me sleeping in the summer. Spread out huge, lose heat from your body so that you stay nice and cool. A counter example is a small surface area to volume. This for cells is not ideal, depending on the cell, I should say. So small surface area means that there will be a minimal exchange with the environment. These organisms here, the whale and the shark, tend to live in cold environments. They're just like fat blobs. They don't have a lot of big appendages, no big ears. Even their flippers and fins are pretty small because they have environmental pressure of cold temperatures. And these organisms need to stay warm. So they should not be exchanging and losing a lot of heat to their environment. So they keep everything more internal to minimize exchange with the environment. Same is true for a lot of organisms that we see that live in cold places. Minimize the exchange. Me sleeping in the cold, curl up in a ball, keep all that heat trapped. So again, minimize exchange. Examples of this are like a neuron. A neuron in your brain tends to have this sort of shape along with like a long tail. We'll get into this more in the coming weeks. But a neuron has to receive and send a ton of signals. So a neuron should have a huge surface area to volume ratio. Something like a cell in your cheek doesn't really have to do a whole lot. It just acts as a barrier. So your cheek cell might look more blobby because it doesn't have to get as much food. It doesn't have to get as much oxygen. It doesn't have to release as much carbon dioxide simply because it's not doing that much. So to conclude, surface area to volume is all about the ratio. It's not just a huge cell is best. It matters how much is on the outside of the cell versus how much is on the inside. So it's all about the ratio. You want to have a huge surface area compared to a small volume. So that is why we see that cells are small. Even though this cell on the right has the biggest surface area, if a tiny little particle had to float to the center of that cell, it would have a heck of a time getting to the center. Whereas here, if we need a particle to float to the center is a lot closer. So all about the ratio. You want to have a huge surface area compared to the volume. This all has to do with the cell membrane because the cell membrane is what goes around the outside of the cell. So the cell membrane is what separates the cell from its environment. It goes by this name, phospholipid bilayer, because it is made of phosphates. Those phosphates are hydrophilic and they touch the watery environment. Lipid, because there are tails that are hydrophobic. And bilayer, bi meaning two, because the cell membrane is two layers of phospholipids. You can see that the way that the bilayer is arranged, there is a watery environment inside the cell and a watery environment outside of the cell. So the cell has to be arranged, or the membrane has to be arranged so that the phosphates touch the watery section and the very hydrophobic tails are away from the water. So the inner portion of the membrane is hydrophobic. The outer portion touches the watery inside and watery outside. The phospholipid bilayer is also said to be fluid. Fluid not meaning like it's a liquid, but fluid meaning that the phospholipids can move left and right. If I have a particle that needs to get out of the cell, it might squeeze through those phospholipids. So fluid means that the phospholipids can move a little bit left and right, like a little bit of a jiggle to move out of the way so that particles can squeeze through there. Also in the cell membrane, there are many proteins, channels, pumps, and receptors. So they say that the membrane is kind of like a mosaic, meaning it's made of all these different pieces. Some particles might be too big to just squeeze through those phospholipids left and right. Like a sugar molecule, glucose is huge. Those phospholipids cannot move enough left to right to allow glucose to come in. So they would need some kind of channel that would allow them to go in. Also, cells have pumps in their membrane. Sometimes particles don't just float the way that the cell wants them to go. So the cell has to physically spend energy and pump things in or out. Also, we talked about quorum sensing and how bacteria communicate. Well, all cells pretty much communicate with those that are around them. So cells have receptors in them where molecules can bind and the cell knows then some kind of communication. If you think again about a neuron, the way that a neuron fires is that a molecule binds to a receptor and sends a signal through the cell. More on neurons in the coming chapters. But big picture here is that the membrane is not a cement wall. Things can pass through in and out of the membrane, and then also the membrane is made of phospholipids, but also proteins that are in those phospholipids. Continuing on, how does a cell allow things in and out? There's passive and active transport. Passive transport does not require energy, meaning particles are floating from high to low concentration. This is an idea we saw in cellular respiration and photosynthesis. Particles move from where there's more of them to where there's less of them. We saw that in respiration with hydrogen ions. The cell set up a scenario in the mitochondria where all these hydrogens were congregated together on one side of the membrane. Those hydrogen atoms then spread away from each other and they flowed through a protein called ATP synthase. And when those hydrogens flowed through ATP synthase, it caused the production of ATP. So we have this idea of a concentration gradient where particles will spread out from where there's more of them to where there is less of them. That is diffusion. So diffusion is particles moving from where they're more concentrated to less. That will occur until equilibrium is reached meaning there's an equal concentration on both sides. That's shown in these pictures on the right. Here I have a gradient. All of my molecules are on one side of the membrane. A law of thermodynamics says particles spread out. So those molecules are going to spread out. Here they go. They're moving across the membrane until there is an equal number on each side. At that point, there is no concentration gradient. There is no congregation of the same thing on one side. So particles then are moving freely back and forth. Same is shown on the bottom pic. We have a concentration of purple on the left and a concentration of orange on the right. They will spread away from like particles until there is an equal concentration of both on both sides of the membrane. One example of passive transport is osmosis. So osmosis is diffusion, meaning that it is passive and energy is not required. Osmosis is the movement of water. Shown here is a permeable membrane, and it appears that that membrane is permeable only to water because I do not see the purple particles moving. So it is permeable to water only. Water will spread from high concentration to low, meaning from where there is more to where there is less. If I look compared to the purple particles, the concentration of water is higher here on the left. The ratio of water to purple is higher. So water will spread from where there is more water to where there is less water until the concentration of water is the same. It's not the amount of water is the same, it's the concentration, meaning relative to the other particles. There's 3 situations a cell might find itself in. In all of these situations, water will move from where there is more to where there is less water. Situation 1, hypertonic. That prefix hyper means more. Tonic refers to the saltiness of a solution. So hypertonic is saying that there is a more salty solution. So here we have in the diagram the inside of the cell, represented in this little dish, the outside of the cell, and the solution the cell is in is hypertonic. So my environment is more salty. More salty meaning less watery. So water will flow from where there's more water to less. This cell will lose water. That will result in the shape of the cell being disrupted. Here you see a blood cell that's normally plump and fat has lost water, so the cell is shriveling up. A plant cell shown below also has shriveled up. The inside here is normally pressed around the outside, but it has shriveled up. So if I watered a plant with salt water, my plant cells would shrivel up because the plant cell would lose water to the salty environment that I created in the pot. Another example of this is like a pickle. A pickle is made from a cucumber. If I put a cucumber into a jar and I put a ton of salt into that jar, which is how they make pickles, from cucumbers and salt, that cucumber is going to lose huge amounts of water and thus create what we know as a pickle. Ideally, a cell will not want to be losing massive amounts of water, nor would it want to be gaining massive amounts of water. So generally, the best situation a cell can find itself in is an isotonic environment. An isotonic is equally as salty, so water will flow equally in and out of the cell. Here you see a normal red blood cell in an isotonic environment. If you've ever gotten an IV or you know much about IVs, An IV that you get at the hospital when you're very dehydrated is not pure water. It is a salt solution, a saline solution that perfectly matches the concentration of salt that's found in blood. So it would not be good to give pure water to your blood cells or extremely salty water, but rather an isotonic solution is best. Isotonic solutions also come into play when you think about fish and keeping an aquarium. People who spend a lot of money on fancy fish tanks and expensive tanks, like if you've ever watched Tanked on Animal Planet, huge money is spent. Those people hire somebody to come into their house to make sure that their fish tank is always kept at the right saltiness. Because if you put a fish into too salty or not salty enough water, it's going to screw up the cells and potentially kill the fish. So fish, like all cells, need to be kept in an isotonic environment. The third situation a cell can find itself in is a hypotonic solution. A hypotonic solution is less salty. So the cell is very salty. The environment is less salty. So water flows toward the salt. Water flows from more water to less water. Water is rushing into the cell. This can potentially cause the cell to burst or explode. So here we have cells pictured on the right. It's kind of hard to tell without a comparison pic, but water is rushing into those cells, making the cells bigger, inflated, and they can potentially burst. One example of this is like some grocery stores have sprayers above the produce. So if I have, let's say, like a little tomato, and I'm thinking about buying that tomato, it's advantageous for the grocery store to put water onto the tomato. So water goes into the tomato, makes the tomato look plump and juicy, and then me as the consumer might be more likely to buy a plump, juicy tomato. So it's creating a hypotonic environment so that water rushes into the cells. To conclude here, there's three situations the cell can find itself in. One in which the cell gains water, water flows in. One in which the cell loses water, water flows out. And one in which water is moving equally and out of the cell. Isotonic would be best for the health of a cell.